A very good morning to everyone. My topic of presentation is a new age dental implant. Looking into the overview of dental implants, the implant dentistry is the second oldest discipline in dentistry, which ages back 4,000 years ago. In ancient Chinese was where the first implant was placed and used. And based on the material, looking into the history, it was only in 1900s the materials like aluminium, gold, and nickel were used in dental implants. And only in 1938 that cobalt, chromium, molybdenum alloy was brought into the placement of dental implants. Looking into the importance of the dental implants, it is closest to that of the natural teeth, and they can be fixed permanently and allow people to chew without any problems. And it has got better aesthetics as compared to other forms of tooth replacement, and thus patient regains confidence. Initially, the process was as a two-piece implants, and the recent trend is to use a one-piece implant. The concept of osseointegration was given by Brainmark in 1977, which is the direct contact of the living bone with the surface of the implant at the light microscopic level of magnification. So there are certain shortcomings of an osseointegrated implant. So this is the comparison between the natural tooth and an implant. The implant will lack the periodontal ligament and it also lacks proprioception. And when there is a large bone defects, definitely it necessitates bone reconstruction before implant fixture. And orthodontic movement is not possible on dental implants. So this could result in a gingival recession due to modified tissue architecture, or if there is a presence of any localized bone loss around the implant fixture, then it would require surgical intervention. To correct any large tissue defects, we need to regenerate the damaged tissues. So, relatively new and highly promising field of reconstructive biology that draws on the recent advances in medicine and surgery and molecular and cellular biology and polymer chemistry and physiology is called as a tissue engineering. Now, tissue engineering is driving the biotechnology train of the future. A new and ambitious approach is that it combines the knowledge for the material chemistry with the cell biology and medicine. This uses biodegradable polymers to make scaffolds into the cells and then into the tissues in the presence of growth factor. The first generation tissue engineering used for constructing cartilages or bone, and the second generation requires a new method of tissue reconstruction not based on the scaffolds. Looking into the importance of periodontal ligament, this provides the progenitor cells for the alveolar bone formation and remodeling. At the bone site facing the tooth root, the PTLs plays the role of the periosteum. The function of the periodontal ligament induces bone even at an ectopic site. Now, PTL expresses its own suppresses its own ossification and possesses a capacity for spontaneous regeneration during which the biochemical strength is restored and innovation is re-established. Now, periodontal ligament cells is a cue for regeneration. This forms a natural connection between the tooth root and alveolar bone and the gingiva. But several biochemical or biomechanical characteristics that osseointegrated implants do not have is the flexibility, proprioception, and physiological tissue remodeling. Now, this is all because it lacks the periodontal ligament. Now, tissue can be destroyed and rebuilt in 3 weeks, but the periodontal ligament progenitor cells, when kept on a new layer of cementum attached to original cementum, it forms a new transverse fibers which get integrated into them. If new cementum layer were to be laid down on the surface of an engineered devices, this would accommodate periodontal ligament attachment with the potential to stimulate the regeneration of adjacent alveolar bone. An implant system that would include a periodontal ligament with the tissue inducing property. That is, a periodontal ligament cells impregnated implant biomaterial is called as a Ligaplant, which is what we are going to be discussing about. Now, any osseointegrated implant will have several hurdles. That is, it cannot be placed in bone defects or when there is a poor bone quality, and it can also have certain complications like bone loss and gingival recessions around the implants. Now, to overcome all these hurdles, this led to the concept of tissue engineered implants. Now, here the materials and the methods which are used for ligaplants is that first you need to prepare a temperature responsive culture dish and then do the cell culture and the culture of the periodontal ligament cells in a bioreactor. So, first will be the preparation of the temperature responsive culture dishes. Now, this is done by N isopropyl acylamide monomer in 2 propanolol solution spread onto polystyrene culture dishes. Now, the temperature responsive polymer grafted dishes is rinsed with cold water to remove the ungrafted monomer and sterilized with ethylene oxide. Next step will be the cell culture. So, from an extracted tooth, the PDL cells is scraped from the middle third of the root and placed into the culture dishes which contain the specific essential medium. And to harvest the sheet, the human periodontal ligament cells were plated on the temperature responsive culture dishes to obtain a cell density of 1 in 10 power 5. Now, then followed by the culture of the periodontal ligament cells in a bioreactor. So, titanium pin is coated with the hydroxyapatite and placed in a hollow plastic cylinder, leaving a gap of 3 mm around the pin. Now, a culture medium was continuously pumped through this gap. So, a single cell suspension obtained from the human were seated first into the plastic vessels under a flow of growth medium for 18 days. This is how a Ligaplant was being produced. Now, let's look into the literature support on the Ligaplants. Now, Gold P in 2010 did a very characteristic experiment where the cells were isolated from the PDL of humans and dog models cultured in a bioreactor, and pins were implanted in a enlarged dental alveoli. Now, histological examination on the dog model shows that the cells were arranged in a typical ligament like fashion. And in human patients, the probing and the mobility assessment suggested that the implants were well integrated with mechanical properties similar to those of the teeth. So it was concluded that the cell covered implants integrated without any adverse effect and induced bone in their vicinity. The new tissue consistent with the PDL developed on the surface of the dental implant after implantation, and this proof of the principal investigation demonstrate the application of the ligament anchored implant which have potential advantage over osseointegrated oral implants. Now, advantage of this ligament implant over conventional dental implant is that in an osseointegrated implant, it lacks PDL, and but it has a concept of osseointegration, but it has only mechanoperception. It lacks proprioception, and bone defects requires bone reconstruction before implantation. If there is a localized one, then the localized bone loss could occur around implants, and also gingival recession also can occur. But whereas in case of liver plants, there is presence of PDL, so firmly integrated without interlocking or direct bone contact, and it mimics natural tooth roots in alveolar bone and can be applied in places of bony defects, induce bone formation, allows movement of ligaplant inside the bone, suggesting intact tissue communication between the bone and the implant surface. So the current shortcomings is that it requires this temperature-induced dishes and the duration of the cells obtained for culturing, and it is at a very higher cost. Any problem during the culturing evokes non-periodontal ligament cells, that is, a fibrous attachment is possible. So hence, I would like to conclude by saying that definitely this Ligaplant, which is a ligament enforced implant, will form a futuristic approach to replace a missing teeth as similar to that of a natural tooth. Thank you for this opportunity and thank you for the patient listening.